Welcome to the Neurology Live Mind Moments podcast. Tune in to hear leaders in neurology sound off on topics that impact your clinical practice. I'm your host, Marco Meglio, editor with Neurology Live. In this episode of Mind Moments, Dr. Sarah Hoffmann, a senior neurologist at Charité University Medicine in Berlin, discussed the clinical challenges and evolving understanding of seronegative generalized myasthenia gravis. In the discussion, she outlines how the condition is defined in the absence of detectable antibodies and why it remains a heterogeneous and often difficult to diagnose population. Hoffmann also reviewed key diagnostic hurdles, including limitations of antibody testing and electrophysiology, and highlighted how these challenges impact treatment decisions and clinical trial development. She also discussed the need for biomarkers to better identify patients and the importance of reassessing diagnosis in those who do not respond to therapy. Let's dive in.
So the name already says it. Seronegative MG is defined by the absence of detectable serum autoantibodies that typically are associated with MG, such as AChR antibodies or anti-MuSK and LRP4 antibodies. I would like to say and mention that the status of LRP4 antibodies is still under discussion, and it's a big debate in the MG community because their role in the pathogenesis of MG isn't fully established. And in terms of prevalence, seronegative MG accounts for roughly 8 to 15% of the overall MG population. And this can vary depending on the different epidemiological studies there are out there.
Well, obviously, we're still learning a lot more about this population specifically. So let's talk about diagnosis, which has been a little bit of a challenge sometimes for clinicians. So, from a diagnostic standpoint, what are the greatest challenges neurologists face when seeing a patient suspected of having seronegative MG?
Yeah, so there are two hearts beating in my chest. This is how we would say it in German. So sometimes it's very, very easy. So there are some very typical symptoms in MG, such as asymmetrical ptosis or a severe bulbar or fluctuating bulbar syndrome that sometimes makes it very, very easy to diagnose seronegative MG. In those patients, I actually don't even wait for the antibody tests to come back because I can be clinically sure. And we do actually see this, especially in the late onset, mainly ocular population, that they can be seronegative in around about 50% of cases. What's actually more challenging are the patients that have a predominant affection of the limbs, because this can sometimes be difficult to differentiate from other neuromuscular or even functional disorders. And from a diagnostic standpoint, we know that autoantibody testing is this cornerstone in clinical practice. But when these results come back negative, it's getting challenging. The second and next most important diagnostic pillar is electrophysiological testing, namely repetitive nerve stimulation and single fiber EMG. These tests, however, do require significant expertise, and especially single fiber EMG is not universally available in all of the sites. And of course, even with experienced examiners, we know that from all diagnostic tests, none of the tests will ever have a 100% sensitivity. So the third diagnostic pillar would be pharmacological testing with acetylcholine esterase inhibitors. And when you have a very clear and visible symptoms such as asymmetric ptosis, and this improves with acetylcholine esterase inhibitors, you have the diagnosis. But again, coming back to those patients with predominant limb affection, they can say, yeah, I'm feeling better with pyridostigmine, but this can be hope or expectation. We know that from now all the nice and good and high quality phase 3 clinical trials, so we do see an impressive amount of placebo response, actually. So this alone cannot, especially when the clinical syndrome is not crystal clear, cannot establish the diagnosis of MG. And this is where we're at. This is the challenge with seronegative MG.
Oh, great, great breakdown. And obviously, there's some things that might be a little bit more complementary towards the evaluation when seeing a patient with suspected seronegative GMG in front of you. But really, really great breakdown, Dr. Hoffman. Kind of encompasses all of the different things that clinicians should understand when a patient does present to them in front of them. So let's talk a little bit about treatments. Obviously, we're still waiting to hear the decision on efgartigimod for seronegative GMG, but what has been the typical approach for treating these patients? And is there variation to the approach based on institution and based on clinician themselves? Talk a little bit about the treatment paradigm per se.
So in terms of basic therapy, the treatment for seronegative MG does not differ from antibody-positive MG. So we rely on acetylcholine esterase inhibitors for symptomatic treatment, and then followed by immunosuppression with corticosteroids and first-line immunosuppressants like azathioprine. However, we are now entering an era, and this is what you're alluding to, where antibody status not only aids in diagnosis, but also guides treatment decisions. And for AChR and MuSK antibody-positive MG, we now have targeted therapies at hand, such as complement inhibitors for AChR positive MG and FcRn inhibitors for AChR and MuSK antibody-positive MG. And for seronegative MG, these therapies are still off-label. They are not yet available for the broader patient group. But of course, this can be used in individual cases. And for example, FcRn inhibitors such as efgartigimod are on label in Japan, and they are used in seronegative and LRP4 positive MG patients in Japan. We cannot do that due to the EMA and FDA approval in the States and Europe.
We're going to take a quick break from our conversation with Dr. Hoffmann to keep you up to date on the latest developments in neurology. Welcome to the Neurology News Minute. Here's what you may have missed since our last episode. First up, on March 23rd, the FDA accepted Ionis Pharmaceuticals' new drug application for its investigational RNA-targeted therapy, zilganersen, as a potential treatment of patients with Alexander disease, a rare neurologic disorder. The agency has assigned a PDUFA target date of September 22nd, 2026 for a decision to be made. 2 days later, the FDA granted accelerated approval to Denali Therapeutics' Tafenoquis alfa as a treatment for neurologic manifestations in pediatric patients with Hunter syndrome. Marketed as Avalaya, it becomes the first therapy specifically engineered to cross the blood-brain barrier for this rare lysosomal storage disorder. And lastly, on March 30th, the FDA approved a new higher dose strength for nusinersen as a treatment for spinal muscular atrophy. This new dosing regimen, which is comprised of 50 mg, 50 ml, and 28 mg, 5 ml doses, is designed to deliver a higher concentration of the drug through both the loading and maintenance dosing phases. For more information about these stories, head to neurologylive.com, or you can check out the corresponding links in the show notes. And now, back to our conversation with Dr. Hoffmann.
That's interesting. I hadn't even known that it was already approved in Japan. So obviously we'll see. And I know we're waiting that decision that'll come hopefully within the next couple of months, and add another treatment, an actual treatment specific towards seronegative GMG. So obviously, Dr. Hoffmann, you as yourself, who's been a big name in the research realm, you've obviously led clinical trials yourself. How do we kind of facilitate more research and more clinical trials geared towards seronegative GMG? Just talk a little bit about some of the challenges that we've had and maybe how we can potentially kind of get the ball rolling a little bit more in this area of research.
Yeah, I think this is still a very complex and difficult topic, and we have seen that in the ADAPT-CERON trial. So I'm actually very thankful for that trial, because it is a trial that is dedicated to AChRB negative MG, but this included MuSK antibody positive, LRP4 antibody positive, and triple seronegative MG. So the first results are out, and in the overall population, we do see positive results. But if you look at the subgroups, it again becomes complex. So I think the biggest challenge we alluded to that a bit. It's a heterogeneous group, and there will be real seronegative MG patients within that group, but there will also be patients with other diagnoses. And I think the main goal for us all would be to find, best case scenario, a biomarker that could differentiate between the two. I mean, we still have to rely on our clinical view and also the electrophysiological testing, but we've talked about the limitations of that. And the biomarker would, of course, be the ultimate goal. And I know that internationally, globally, multiple research groups are on the hunt for the next autoantibody. We at our study site, what we do is in individual cases, we actually perform intercostal muscle biopsies together with our thoracic surgeon. It's a flat muscle with abundant neuromuscular junctions, and we look for complement and IgG deposition in selected cases. So this could be one approach, but of course it's an invasive procedure, and it's not going to be done for a number of reasons, including ethical ones in the vast majority of patients. But the challenge that I see, because I'm so deeply convinced that seronegative MG exists, I've seen it multiple times, that if we are talking about especially therapeutic trials, we really, really need to stratify and select the patients in order to not dilute the outcome. And this is the biggest challenge. And again, I'm back to the biomarker, but it's still not there.
Yeah, I mean, it's hard getting a homogeneous population for a population that is already heterogeneous to begin with. You talked about biomarkers kind of being the big thing that you'd hope to see kind of come to fruition over the next couple of years. So how close are we to potentially getting to that biomarker? And have we seen any signs of life or any, things that have popped up that we've maybe been promised, have shown a little bit of promise so far?
Yeah. I mean, of course, I'm heavily biased here. When I talk about my intercostal muscle biopsies, I'm convinced that we do have a biomarker there. But of course, we would wish for something like a blood-based biomarker because that's easily accessible. And I do not see that on the horizon yet.
Well, obviously, like you mentioned, there's still a lot of groups that are, working towards it, and hopefully we do start to see a little bit more of that over the coming years. It's been awesome that there's been so much activity in the myasthenia gravis realm over the last decade or so. The fact that we're having this conversation about seronegative treatments and possible precision medicine as well is really, really awesome. And so that kind of brings me to my last question for you, Dr. Hoffmann, and great clinical insights on all of these. I appreciate you taking some time out of your day. So last question, which you've kind of talked about a little bit throughout our conversation, but regarding seronegative GMG, what are some of our most important unanswered questions that we currently still have? And what do you feel as though the clinical community needs to kind of be focusing in on as we go forward in the next couple of years?
Yeah, one part was already mentioned. I think the biomarker is something that we all are focused on and that we should, with the different capacities that we have, with the different methodological approaches, focus on. But I also think from a clinical perspective, if I may answer that non-scientifically, because I do see that a lot in my outpatient clinic. At one point, we have to decide if a patient is a seronegative MG patient or not. But if we do establish that diagnosis, but within the long run, we don't see the patient responding to our usual therapeutic approaches. I think we do have to take a step back again and question our diagnosis. Be brave enough to open that question again. And on the other hand, if a patient, and I've seen that also numerous times, especially in younger female patients, they do get a functional disorder diagnosis and in the end had seronegative MG. And this is the real challenge there, because in those patients, we miss a window of opportunity for hit hard and early immunotherapy. And in the others, we may overtreat and expose patients that cannot benefit from immunotherapy because they have a different diagnosis. We pose potential risks to them. So from a clinical perspective, I think we always have to second guess our diagnosis, especially when we do see non-responders to our therapy.
Thanks for listening to this episode of the Neurology Live Mind Moments podcast. To support the show, be sure to rate, review, and subscribe wherever you listen to podcasts. And as always, for more neurology news and expert-driven content, visit neurologylive.com.